Hello everybody, my name is Daniel Manriquez. I'm post-harvest and plant physiologist, and welcome to this last presentation in this Cuticle Academy second version. And let's start with my presentation. The title of my presentation is Plant Cuticle Enhancement and His Impact in Marketable Yield. First, let's talk a little bit about how important is the cuticle for the plants. Cuticle is a waxy layer that is covering different organs and structures in the plant, fruits, leaves, stem, and even roots, and is the first line of defense providing protection from the outside elements such as sun, rain, and wind. And also the cuticle can regulate the exchange in moisture and air between the plant and the environment, and can act as a barrier against pathogens and insects. For all those reasons, the cuticle is vital and is playing an important role in plant health, fruit quality, and also in optimizing yields. And cuticle structure is based on an epicuticular layer where mainly the waxes are, and a cuticle layer that is internal and in close contact with the cell wall of the epidermal cells. This epicuticular layer is so important because it's the layer that is in contact with the environment, but also with the pathogens and the insects. Let's talk a little bit about Cultiva and why Cultiva is a pioneer in the development of a technology to improve the plant cuticle. Cultiva is a global company with presence in different countries around the world. We have operations in North America, in countries like Canada, US, and Mexico. Also, we have operations in South America, in Chile, in Argentina, and now we are growing in Brazil and other geographies like Peru. Also, we have operations in different countries in the European Union and in NC, like Australia and New Zealand. Why Cultiva is a pioneer in cuticle improvement and plant health? Because we have a solution called Parka. It's a technology developed by Oregon State University years ago, and is a blend of phospholipid and polysaccharides. Both group of compounds, phospholipids and polysaccharides, are normal compounds in plant cuticle. And our Parka technology is, by this way can improve, the natural cuticle of the plants. Let's talk a little bit about the characteristics of Parka. Parka is elastic. It's a coating that show and expand when plants and fruits are growing. Also, there is an important component related to hydrophobicity because of the phospholipids in the formulation. They can provide, a better hydrophobicity to the cuticle of plants. And Parka is permeable. It's not affecting the gas exchange between the plant and the environment. It's not an anti-transparent. Another important characteristic of Parka technology from the logistic point of view is a product that can be mixed with different other compounds in the tank, like insecticides, fungicides, or foliar nutrients. It's edible because all the components in the formulation are food grade. And also it's a clear coating that is not trivial because it's not affecting the appearance of fruits and leaves. And we will see that in one of our slides in the presentation. But let's talk a little bit about the effects of Parka in cuticle improvement. And let's start first looking at the effect of Parka in this external layer of the cuticle, the epicuticular layer. In this experiment, we were working with tomato fruits with the cultivar kernel, and in these images from the epicuticular layer, you can see, the epicuticular layer in control fruits versus fruits treated with Parka, and you can see the deposition of Parka in this epicuticular layer and how this deposition is changing, this, external layer of the cuticle. Looking at the same experiment that was conducted with a group in Spain, led by Eva Dominguez and Antonio Heredia, here we can see, in two tomato varieties, Gardner and Cornell. Gardner is a high cracking susceptibility variety, and Cornell is a low cracking susceptibility variety. And we can see in Parka and in control how the structure of the cuticle is changing across the development and the maturity of the maturity process in both cultivars. And we can see, for example, at breaker stage in tomatoes control and treated with Parka, how the deposition of the cuticle is totally different. You can see differences in the epicuticular layer, but also you can see at cuting level, changes in the structure of the cuticle and in the deposition. Look at the invagination of the cuting in the fruits treated with Parka. You can see how the cuting is involving in some way the epidermal cells, providing a better structure to the fruit cuticle. Now, if we look at the thickness of the cuticle on Gardner tomatoes across different fruit stages of development, here we can see in a very early stage of development, 12 days after antithesis, how fruits treated with Parka are showing a better thickness of the cuticle in comparison to the control. The same thing is possible to see in different maturity stages across, fruit development, where fruits treated with Parka are showing a better thickness in comparison to the fruits coming from plants untreated. Now, if we look at the amount of cuticle deposition in the fruits for the same variety across 3 different maturity stages, and we can see again that fruits treated with Parka are showing, a higher amount of cuticle in comparison to fruits coming from untreated plants. Now, if we look at the components of the cuticle, from one side waxes and from the other side, cuting, we can see how again fruits treated with Parka are showing, a higher deposition of waxes in comparison to fruits coming from untreated plants. Same situation for cuting. The cuting in fruits treated with Parka, the deposition of cuting in fruits treated with Parka is much higher in comparison to fruits coming from untreated plants. But what happened in terms of leaves? Because if we think in an agronomical application of Parka, when we are spraying in the orchard or when we are spraying a plant, a row crop, an important percentage of this product is going to leaves and a small percentage is going to fruits. And let's see what happened when we apply Parka in the field. What happened in terms of cuticle of the leaves? And here in this experiment that was conducted by Wageningen University in Holland, they were using lettuce as a plant model to look at the modification of Parka in the leaves, in the cuticle leaves. And we can see here in the same images, in this column, control leaves, and in this column, Parka treated leaves. And first, we can see the stomatas in both leaves, and we can see that the stomatas are open. I mentioned that at the beginning of my presentation. Parka is not an anti-transparent. It's not inducing a closure of the stomata. The stomatas is still being open when we treat the plants with Parka. And if we look at the cuticle of the leaves in young leaves of lettuce in these images here, you can see that in the untreated leaves, you can see a lot of microfractures, but in the case of Parka, we can see a better cuticle condition in comparison to the untreated leaves. When we go to old leaves, 6 weeks, we can see that the cracking or the micro cracking in those untreated leaves is much higher in comparison to Parka, where you can see a better structure of the cuticle. Now, looking again at thickness of the cuticle in leaves, this is the same experiment that we were talking before, conducted by Eva Dominguez and Antonio Heredia in Malaga University. you can see how the thickness of the leaves in plants treated with Parka is higher in comparison to untreated. And you can see that especially in the abaxial side of the leaves in both varieties, in Gardner, and Cornell. Now looking at the same, you know, improvement in leaves, and this is an example coming from Lerida University. This experiment was conducted by Isabel Lara on apples in the cultivar Fuji, and we can see, now in terms of total waxes, how Parka is improving the amount of waxes in the cuticle leaves of apples in comparison to untreated. The same situation was observed in the case of cutin, where the leaves treated with Parka or trees treated with Parka are showing, a higher deposition of cutin in comparison to untreated. Another important point to consider for this, experiment, this sample was taken during a very hot period. For perhaps you don't know Lerida, where this experiment was conducted in Spain, but Lerida is a very warm area during the summer, and those leaves were collected in the middle of the summer when the temperatures were reaching above 35 degrees Celsius in many days during the month of June and July, and when the relative humidity was pretty low, near 50%, under very stressful conditions for the trees. And we can see this modification on apple leaves. But let's talk a little bit about, agronomical results when we apply Parka in the field. And let's start with fruit quality. And in the case of cracking and micro cracking, there are some scientific papers that are showing that, when the fruits are in early stages of development, 24, 25 days after full bloom, is when, the micro cracking in the fruits start to be developed. And why? Because it's at this period, in the case of cherries, when the fruits are going from, cell division to cell expansions. The fruits are going from growth coming from cell division to cell expansion. And in early stages of development until this period, the epidermal cells, they have the energy and the capacity to synthesize cuticle in an amount that can cover the entire surface of the fruit. But when those fruits are going from this growth from cell division to cell expansion, and we go to an exponential growth, those epidermal cells, they don't have the energy and the capacity to synthesize cuticle to cover the entire fruit. And this is the reason why at those early stages of development is when the fruits, they start to develop cracking. And some other studies, this was conducted at Concepcion University here in Chile. They are showing that there is a direct correlation between microfractures or micro cracking in early stages of development of the fruit and the cracking that you can see at harvest. This is the reason why it's so important to start reinforcing the cuticle in very early stages of development of the fruit, because there is this direct correlation between microfractures development and cracking at harvest. Going to this experiment tomatoes again, when we were working with two different varieties with high and low cracking susceptibility, Gardner and Cornell, we can see that when we apply Parka, and due to this reinforcement in the cuticle, we saw a significant reduction in cracking in the case of Gardner, and also in the case of Cornell, a low cracking susceptibility variety. In both varieties, the reduction in cracking at harvest was reduced by the application of Parka, and is due to this reinforcement of the fruit cuticle. Now looking at some experiments that we conducted in cherries across different seasons, different varieties, if we put those results in a blend and we have an average, we can see a significant reduction of cracking at harvest in different varieties, different seasons, and different geographies. This can show, you know, how solid is the control of cracking when we apply Parka, especially when we are applying Parka in a program starting very early in the season, reinforcing the cuticle to reduce the micro cracking. And by this reason, or because this reason, we can see a significant reduction at harvest. Looking now at results in cracking reduction in other crops, this is blueberry. It's the same exercise. We took different varieties, different geographies, and we calculate the average of reduction in cracking in blueberries. And we can see a significant reduction. This is not trivial. In the case of blueberries, now with the expansion of the production around the globe and where we are growing, blueberries in different growing areas, where perhaps, during the season we have, not rain, sometimes fog could trigger, the cracking development in some varieties, especially in the new varieties that are showing, bigger sizes in comparison to the old varieties. Now looking at other crop, here you can see in the case of table grapes, how Parka is reducing, the amount of micro cracking in two different varieties: Red Globe, a traditional old variety, and Muscat, that is not a common variety for fresh consumption, but the consumers that are looking at this variety because of the flavor. And you can see how Parka is increasing the percentage of some fruits in both varieties, and is reducing the cracking incidence, especially in the case of the distal and the distal microcracking in both varieties. And also we can see that in the case of Red Globe for this experiment, Parka is reducing the incidence of pedicel cracking, microcracking. Let's talk about other problems related, high humidity or rain during the season. This is an experiment that was conducted years ago in the East Coast on Fuji apples, and here we can see the reduction of russet. Russet is, a problem for different varieties of apples, and Fuji is quite sensitive to develop this problem, especially in areas with high humidity or rain during the season. In the case of Brazil, for example, where the apples are growing in the south and they have a lot of rain during the season, the russet incidence could be really, really high at harvest, especially in sensitive varieties like Fuji. But in this graphic, you can see how Parka can reduce the incidence of russet in comparison to the untreated fruits. But also, Parka is showing, a synergy when it's combined with this PER. Provide is a mixture of J4 and 7 that is a normal PER used by the industry to reduce this problem on apples. And Parka is showing a synergy between, this J4 and 7 in russet reduction. Now let's go to peaches. And here we are talking about autumn snow, a white flesh peach. And you can see how Parka is reducing staining in this variety. In general, for peaches and for nectarines, white flesh varieties, they are showing a super high susceptibility to mechanical damage, and staining is a symptom of this mechanical damage. And we can see how Parka applied 3 times or 4 times during the season can reduce in a significant way the incidence of staining in those, white flesh varieties like autumn snow. Let's talk a little bit about plant performance, because as I mentioned at the beginning, the cuticle is critical in terms of plant health and the protection of the plants against, the environment. And this is quite related to the plant performance. And here we can see in this experiment that was conducted years ago in Spain on cherries, where the researchers they were measuring different parameters associated to plant performance, like net CO2 exchange, stomal conductance, transpiration, and SPAD. And you can see in this measure that was conducted the 1st of May in those cherry trees, you can see how Parka is increasing the net CO2 exchange in a significant way. Also, we can see a trend, and Parka treated plants are showing a better stomatal conductance. In terms of transpiration, we can see the same phenomenon that we saw in the net CO2 exchange, and trees treated with Parka are showing a better transpiration. And if we look at the SPAD that is related to the chlorophyll content, we can see that leaves treated with Parka are showing a better SPAD in comparison to untreated. This is another experiment that was conducted in Talca University, in this case on Fuji apples during the season. Remember, December, January, and February is the season for us in the South Hemisphere. And look at those two parameters, again, stomatal conductance and chlorophyll content measured as a SPAD. And we can see that in trees treated with Parka, we have a better stomatal conductance across the season. And also we can see a better chlorophyll content in comparison to untreated leaves. This is a very interesting concept that we like to use at Cultiva for our experiments when we talk about, plant performance, and is water use efficiency. Because water use efficiency is combining from one side, transpiration, photosynthesis, and is correlating in some way that with production, yield increase is how the plant is using the water in a better way, correlated with the photosynthesis. And you can see in this experiment that was conducted on Fuji apples in Spain, and you can see how across the season in this super hot area in Spain, Lerida, you can see how trees treated with Parka are showing a better water use efficiency, especially in July and August, where it's super hot in this region. When we talk about plant performance, of course, we need to talk about yield, because at the end of the day, if we have a better performance, we will have a better yield. And this is a good example. Here we have the good example of plant performance and how Parka, due to this effect in improve plant performance, we can have a better result in terms of yield. And in this graphic we can see three different almonds varieties: Independence, Nonpareil, and Caramel in California. And you can see how Parka applications during the season is providing a result in a yield increase for the three varieties at harvest, measured as not mid-yield, but it's pretty consistent for those three varieties. If we do the same exercise but now on citrus, and looking at this experiment again that was conducted in California years ago, and we were measuring the yield at harvest on oranges treated with Parka across the season, you can see again a clear increase in yield in pounds per acre in the treatment with Parka. Let's talk a little bit now about abiotic stress. All of us, we know that abiotic stress is more common due to this global climate change, and in most of the regions, growing regions, we have very stressful conditions during fruit development. And let's talk about some results in this area when we use Parka in the field. This is an experiment that was conducted on tomatoes. This was this season. And in this experiment, we use, this NDVI measurement that is talking about plant status to look at how the plants are performing in plots untreated and treated with Parka. And when you look at this graph that is showing the NDVI, measurement across the season and the plant development, you can see that at the end of the season, plants treated with Parka are showing a better condition, a better performance in comparison to untreated plants. And you can see the same things in the images. Look at those images that were, taken at the beginning of the cycle, and look at the plots. This was the area treated with Parka, and this was the area untreated, the grower standard. And you can see at the beginning of the season how the area treated with Parka seems to be under stress in comparison to the grower standard. But look at the situation during the season, and especially at the end of the season, those points that are in the graph. Look at the green areas in the plot treated with Parka in comparison to the plot untreated or grower standard. You can see that the areas treated with Parka, we can see bigger, greener area that is showing that plants in this plot are in a better, status. And what happened at harvest? We can see that at harvest, the yield in tons per acre in the area treated with Parka, it was, much higher in comparison to the untreated area. And I apologize because of my camera, because it's not permitting to show, the appearance of the field, but I'm pretty sure that my colleagues, they will share with you the presentation, and in the presentation, you will see clearly in the pictures the differences between both plots, untreated and treated with Parka. This is with Parka, and you can see in this small corner, the area untreated or grower standard. But again, this better plant performance will provide you at the end of the season results like this result, a yield increase when we apply Parka. But stress is not only important during the season. During the season is important for us because, if the plants are under stress, they cannot work in a better way, and by this way, the development of the plant is affected, the development of the fruit is affected. But also, we need to keep in mind that stress is important also in post-harvest. And this is an example in cherries. This is an experiment that was conducted in Chile, and again, we need to change our mindset to South America and South Hemisphere. And let's talk a little bit about the stress during post-harvest, when the trees, they don't have fruits. During this period, for deciduous fruits, like cherries, apples, or pears, stress is critical because in the case of cherries, for example, during this period, immediately after harvest, we have, a flower bud differentiation. And if we have a stress during this period, we can affect in a very negative way this differentiation. And at this point is when, for example, doubles and spores are developed. The doubles and spores that we are looking at in the season are coming from the last season, from this period, from the post-harvest. And mitigate the stress during this period is important in the case of cherries to reduce doubles and spores, to reduce and mitigate this negative stress that can affect, again, differentiation. But also this period is critical for the deciduous fruits because it's during this period when the photosynthesis is supporting the reserves accumulation. And reserves are super important to support the development of flowers, fruits, and leaves next season in the early stages of development, when the leaves are not, or they don't have the capacity to photosynthesize at the beginning. All those all the development of those early stages, flowering, fruit set, initial fruit growth, and initial leaves development and expansion is supported by reserves that we are accumulating during post-harvest. And this was a very nice experiment conducted in Chile by some colleagues years ago. Because what happened now in our cherry season, and especially for early varieties like Santina, this post-harvest period is really long. Because Santina is an early variety, growers, they will harvest early in the season, and because of that, the trees will be in this post-harvest period for months. And what happened in some of our growing areas here in Chile, looking again at this global climate change, season after season, we have more and more, high temperatures during this post-harvest period. That for us is December, January, February, and March. And you can see in this graph how for this season where we conducted the trial, the number of hours with high temperature in comparison to the historical was higher. And for this experiment, we conducted applications after harvest in December, January, and February, and we were measuring after the applications the reserve accumulation and also the season after in December, November, December. Next year, we were evaluating fruit quality and some other parameters related to production. Let's start first with the plant status during this post-harvest period. Remember, we were applying Parka in post-harvest. It was December, January, and February. And during this period, until March, we were measuring different parameters related to plant status. And in this graph, you can see stomatal conductance. And you can see how plants treated with Parka, they were showing a better stomatal conductance in comparison to untreated plants. When we look now at the chlorophyll content in the same period, December, January, and March, you can see that plants treated with Parka were showing a better chlorophyll content in comparison with untreated plants. Two important parameters to evaluate plant performance, and two important parameters related to photosynthesis and reserve accumulations. But let's start first with fruit quality. When we were to next season to evaluate doubles and spores, we saw a significant reduction in doubles and this kind of fruits that we call here in Chile with sutures. Those fruits, again, this problem is coming from the previous season, and we can see that with this mitigation of the stress that Parka is doing in treated plants, we reduce in a significant percentage the incidence of doubles and suture fruits in the season after. But I was mentioned to you that during this period, during these post-harvest periods in deciduous fruits, including cherry trees, is an important period for the trees to do a good photosynthesis and to accumulate reserves. And the researchers in this experiment, they were conducting, reserves accumulation evaluation in terms of arginine and starch in different, structures in the plant, in the canopy, in the spores, and also in the roots. And you can see that in the case of spores, Parka to the trees are showing, a higher level of arginine and starch. But when you go to the roots and you measure arginine and starch, you can see how Parka is affecting in a very significant and positive way the accumulation of arginine and starch, in comparison to the trees untreated. We were measuring also the season after, not only fruit quality and the incidence of doubles and spores, also we were measuring at, the productivity and also the size. For us here in Chile, size is really important today for cherries because our main market is China, and 95% of our cherries are traveling to China. And consumers in China, they really like big size fruits. And you can see in the size distribution curve where black is untreated and blue is Parka, you can see how Parka treated trees the season after during post-harvest are showing, a higher percentage of fruits in those bigger categories. Look at that in this, 32 to 34 millimeters. Parka is showing a significant increase in the percentage of fruits in this category, similar to this other, category of big size fruits. But which is our theory? Why we think that Parka is moving this curve to higher size fruits categories is due to the reserve accumulation, because remember, cell division is so important to define which will be the final size of the fruit. If we have more cells and the second, the second period of development related to cell expansion, if we have more cells in the second period of cell expansion, each of those cells will grow, and because of that, we'll have, a bigger fruit. The number of cells is critical for reaching this kind of results. And this is the reason why in our mind, in our theory, we are thinking that Parka is affecting. Because if we think that trees treated with Parka, they can accumulate more reserves, they have more energy to wake up during the spring. And the trees, they will have more energy to support processes like cell division. And by this way, we'll have, a better handicap to reach, bigger sizes and harvest. But it's an area where we are working. Now we are conducting a lot of trials in Brazil on table grapes, looking at reserve accumulation. But let's talk a little bit about, stress reduction in the case of fruits. This was an experiment conducted on apples in two varieties in Spain by IRTA years ago. And let's look at those Granny Smith apples during the season. They were measuring, hydrogen peroxide. That is a good, stress marker. And they were measuring the concentration of, hydrogen peroxide during the season. And here we have days after full bloom for Granny Smith. And you can see the concentration of hydrogen peroxides in fruits treated with Parka versus fruits untreated. And you can see the reduction in this stress marker, showing that the fruits treated with Parka are suffering less stress in comparison to fruits untreated. Another expression of stress and is sunburn, and this is a graph showing a sunburn reduction, again for different varieties, different geographies, different seasons. And here we can see how Parka, in the case of apples, is showing a significant reduction in sunburn incidence for all those varieties, different geographies, and different seasons. This is another example showing how Parka can reduce sunburn. This is an experiment that was conducted in California on Caracara, Sumo, and Clementine citrus. And again, you can see how consistent is the reduction of sunburn in those, citrus crops when Parka is applied. And this is the appearance. Do you remember that at the beginning we were talking about, Parka and as a transparent coating? Here you can see leaves appearance and fruit appearance treated with Parka, and those leaves were treated, or those trees were treated with calcium carbonate. And look at the fruit appearance. For the guys that work in post-harvest, this is a nightmare, because try to clean this white stuff, especially in the pedicel area, is complicated. And for export countries like Chile, is really critical because when you arrive to the final consumer and you have a white residue in the pedicel area, in their mind, they will associate this white residue to toxic thing that perhaps can, affect in a negative way their health. They don't like to see white residues in their fruits. And trust me, I'm a post-harvest guy, and clean that in the packing line, even if we use a lot of detergents, cleaning stuff, is not simple. And but again, you can see how Parka is not affecting the fruit appearance, but is helping to reduce sunburn. This is a very nice way to look at how Parka is reducing stress also. Here we are talking about new plantings, baby trees. Baby trees, they are quite sensitive to stress because they are babies, and you can see in those two graphs that are showing, low shoots length and high shoot length, how Parka is improving the development of the shoots across the season, providing to this plant the capacity to have a better structure and a better establishment in the orchard. But again, those are baby trees and eye sensitive like babies. And we need to take care of those trees. And Park is a good tool to protect them against stress and to provide them the capacity to grow in a faster way and to be established in a better way in the orchard. This is another way to look at the effect of Parka in new plantings. This is a measurement related to apex definition. Because when the plants they are in stress and they stop to growth, the first expression is to stop, the growth of the apex area. And again, apologize because my camera is covering, the picture, but you can see in the graph how Parka is increasing the number of, undefined apex in plants treated, providing again, the possibility to the plant to growth and to have, a better establishment in the field. This is a very nice experiment conducted years ago in California. And why is a nice experiment? Because every time in our mind, when we think in stress, we think in water stress, we think in heat stress, but sometimes we don't think that cold is an stress too for the plants. And this was an experiment conducted in citrus. And look at those plants. Those plants, they were in a condition where, cold stress was not induced. And those plants were under cold stress, low temperatures. And look first at the effect of the cold stress in those citrus plants. You can see how the citrus are reducing the development due to the cold stress. But look at the plants treated with Parka. Look at this plant treated 2 times with Parka, or twice, sorry, in comparison to the control under cold stress. And you can see, the better development of the plant and the performance in comparison to the control. And you can see how the development of this plant was similar to the development of a control plant in conditions without cold stress. Nice. This is nice because, when you talk about citrus, and you know much better than me, we're exploring new growing areas where sometimes the temperatures are not the best temperatures to support, the development of the plants. And why not consider Parka as a tool to mitigate this cold stress in these new areas for, the citrus industry expansion. And let me go to some slides to show how, pre-harvest applications of Parka and a pre-harvest, improvement in the cuticle can go to the post-harvest area. I know that you had a presentation before my presentation talking about that. but I want to show some results. This was an experiment conducted on blueberries, and the researchers, they were measuring dehydration during cold storage, 10 days cold storage. Dehydration is something important at consumer level, because when you look when you have dehydration that is reaching, or level of dehydration that is reaching, let's say 4 or 5% of the original weight, you can see symptoms. But symptoms are not the only problem. In some crops, water loss is related to your turgency and firmness. But in the case of this experiment on blueberries, you can see that Parka is reducing the fruits that are showing dehydration in comparison to the untreated, proving again that a reinforcement of the cuticle in pre-harvest can have an impact in post-harvest and even on consumer level after a cold storage period. This is another experiment now conducted on table grapes, and we saw the same thing. This experiment was conducted this year in Brazil in a sugar crisp, a new table grape variety, high value variety. And the colleagues in this experiment, they put those grapes for 15, 30, and 45 days in cold storage, and they evaluate, the dehydration. And you can see how Parka is reducing the berry dehydration in comparison to fruits coming from untreated plants, but especially in long cold storage periods. That for some geographies is important, especially when you are working in exportation, or when you want to store table grapes for a while, looking at a better market condition. This is a disorder that can happen in table grapes, and is related to microfractures. The name of this problem is bleaching, and is this decoloration that you can see in red or black varieties, and it's related to the SO2 that we use in post-harvest to control decay. When you have microfractures in the berries, this SO2 will go into the tissue, will react with the anthocyanins, that are the compounds that are doing, the red colors in the berries, and cause this decoloration. And look at how Parka is reducing the bleaching after 46 days at 0 degrees Celsius, and also during shelf life. Shelf life is the picture that the final consumer will have, and we are reducing, the incidence of bleaching with Parka applications. But remember, Parka was applied in pre-harvest, and we started again at very early stages of development of the berries. And we are reducing the microcracking. And this is the reason why we have a reduction in bleaching after cold storage. Nice. Also, when you look at secondary structures, this is, rachis appearance on grapes, table grapes, and you can see how Parka is improving, the rachis appearance, or is reducing, is reducing the browning in the rachis. Rachis is a totally different structure. It's like the pedicel for the cherries. We call that in post-harvest a secondary structure. And when we are dealing with, post-harvest in the case of table grapes and in the case of cherries, we need to deal with two different structures, the berries, and also those secondary structures that are, in the case of grapes, rachis, and in the case of cherries, pedicels. In the case of the rachis, there are some scientific information that is showing that rachis, they have a higher respiration in comparison to berries. Rachis, they have a lower amount of waxes in the cuticle in comparison to berries. Rachis, they have more stomatas and lenticels in comparison to the berries. This is the reason why rachis is an structure that will lose water, or more water, in comparison to the berries. And but when you apply Parka, and when you apply Parka in early stages of development of bunch, when the rachis is exposed because the berries are small, Parka is reaching the rachis and is improving the cuticle of the rachis. And this is the reason why in post-harvest you can see a better rachis appearance in bunches treated with Parka. It's because we are improving, the cuticle, and we are reducing water loss. This is an experiment that we conducted on cherries, and you can see the same effect. The applications of Parka in pre-harvest are reinforcing the cuticle of this secondary structure that is, designed to lose water. Rachis and pedicels, they were designed to lose water because all the characteristics that I mentioned before. But Parka will improve, their cuticle, and by this way will reduce the water loss during post-harvest, and by this way we can keep all the growers and exporters, they can have, table grapes and, cherries with better rachis and pedicel appearance. Looking at apples now, this is lenticule breakdown. Years ago, I can remember a professor from Washington State University. He was working a lot in cuticle and he was working a lot in waxes deposition in apple cuticles, looking at, the deposition and the conditions that can improve, this deposition in relationship with this problem, with lenticule breakdown. All of us, we work in post-harvest on apples. We know that lenticule breakdown is a serious issue, especially for, some varieties. And this was an experiment conducted, in the East Coast some years ago, or 1 year ago, looking at lenticule breakdown in Honeycrisp. And you can see how Parka applications are showing a significant reduction in lenticule breakdown in comparison to untreated fruits. And again, this reinforcement of the cuticle in pre-harvest is showing, a positive effect in post-harvest. And this is my last slide. And again, apologize for the camera that is, affecting, the presentation. But it's a summary of all the slides that we were going through, and how the plant cuticle is so important and will define marketable yield, and how a cuticle enhancement from one side in fruits, from one side in leaf, will help plants to reduce stress mitigation, will improve plant performance, and will improve fruit quality at harvest and post-harvest. And again, this is a summary showing, it's a summary of our results across different geographies, different crops, different seasons, where we can see how Parka is improving from one side associated to fruit quality at harvest and post-harvest, packouts, and we can have better packouts. And in the other side, related to plant performance, how Parka can help, can be a tool to improve yield. And at the end of the day, provide to the growers and exporters a better marketable yield. And this is my last slide. And thank you very much again for your attention. Every time it's complicated to speak at the end of the season because, people is tired. But it was a pleasure for me to be with you. My apologies for not being present with you at this Cuticle Academy second version organized by Cultiva. But I need to stay here in my country in Chile because there are some activities associated to the family, because my oldest son will be graduated this year, and this is the reason why I'm not with you over there. But all the best. Enjoy the rest of the day. Enjoy the conversations with the colleagues. You have very good speakers in front of you, and use this opportunity to talk with them, to use their science, their knowledge. And also I want to thank Cultiva's team, especially Andrea Velasquez and Victoria Towels, because all the help in that they provide me to prepare this presentation. And again, thank you very much. Enjoy the rest of the day. enjoy Las Vegas. Thank you. Bye bye.